Good afternoon, everyone. My name is Donghoon Lee. I'm an assistant professor at Mount Sinai. I'm truly excited to showcase our depth and breadth of our work with PsychAD Consortium. This initiative represents a monumental effort to build one of the largest single-cell transcriptomic atlas of human brain disorders. I have nothing to disclose. Recent genetic studies have revealed substantial genetic correlation among multiple complex neurological diseases. This means that the same underlying genetic factors can contribute to risk of multiple distinct disorders. Beyond genetics, there is also a significant clinical overlap of these traits. A compelling example is Alzheimer's disease. It's estimated that more than 80% of the patients with the AD dementia experience at least one or more types of neuropsychiatric symptoms that significantly impact their clinical outcome. Our research is aimed at addressing the critical need in neuroscience, which is understanding complex interplay between different brain disorders. Given this genetic and clinical overlap, characterizing both shared and distinct transcriptomic changes across multiple neurological disease has profound implications for early treatments and also development of effective therapeutics. We've assembled one of the largest single-cell cross-disorder dataset to date to uncover shared transcriptomic vulnerabilities across multiple brain disorders. It covers nearly 1,500 unique brain donors. We focus specifically on a dorsolateral prefrontal cortex, or DLPFC, a region that is critical for cognitive functions. Using single-nucleus RNA-seq, we meticulously characterize both cell type composition and gene expression changes that occur in a range of human brain disorders. What makes PsychAD truly remarkable is its comprehensive scope. Our cohort includes individuals with diverse genetic background, spans entire human lifespan, and has a balanced 50-50 split between males and females. This rich data set allows us to gain a truly holistic understanding of human brain and disease. To generate our comprehensive data set, each brain donor underwent 2 technical replicates, resulting in almost 3,000 single-cell libraries. Following a rigorous quality control processes, we successfully generated high-resolution single-cell profile from a 6.3 million nuclei. This extensive data set provides a foundation for our detailed analyses. Next, we annotated these cells using a unified taxonomy specifically for human prefrontal cortex. The taxonomy is structured into 3 hierarchical layers. To achieve this, we employed a series of iterative processes, subsetting, reclustering, and utilizing unique set of highly variable genes at each layer. This allowed us to precisely identify and characterize distinct cell population within the DLPFC. We know population-level gene expression variation is shaped by a multitude of factors. These include individual differences like age, sex, and ancestry. Also, technical factors like tissue source, library prep, and even dissection biases. To systematically dissect the transcriptional variation, we performed variance partition analysis. Our primary goal was to precisely identify the major determinant of expression variations within our data set. Our analysis revealed approximately 50% of the observed expression is coming from differences in the cell type. This highlights profound influence on a cellular heterogeneity. And interestingly enough, we found that 10% of the variation is attributable to individual differences. And this inter-individual variation often represents significant biological insights. A striking example of this was the gene ARL17B located within a critical MAPT locus of chromosome 17. This gene, about 80% of expression variation stems specifically from inter-individual variation. And further investigation into these genes with high inter-individual variability revealed they are enriched with the basal cellular functions. Next, we shifted our focus to cross-disorder comparisons. We concentrate on a set of 8 neurodegenerative and neuropsychiatric disorders that are well represented within our PsychAD cohort. The initial step involved comparing cell type compositional variation for each disorder group against the shared control groups. This allowed us to identify deviation of healthy brain cell proportions. We observed disease within similar category of disease tends to share a similar pattern of compositional variations. For example, many neurodegenerative disorders show loss of excitatory neurons and increased proportion of interneurons and vascular cell types. In contrast, most neuropsychiatric disorders primarily exhibit increase in neuronal cells. These distinct patterns offer crucial insight into cellular pathology of these different disease categories. To better understand both shared and distinct transcriptomic changes occurring across 8 disorders, we employed a Bayesian analytical approach which allows us to evaluate the specificity of disease effects. This framework partitions disease effect into shared and disease-specific components. Our analysis revealed the genes exhibiting cross-disorder signatures were primarily involved with the fundamental cellular processes like mRNA splicing. This suggests a common underlying vulnerability in a basic cellular machinery. After identifying and accounting for these shared signatures, we then used residual disease-specific effect to quantify pairwise transcriptomic similarity between different disorders. Through this, we uncovered a striking transcriptomic similarity between AD and other related dementia, including Lewy body dementia, vascular dementia, and Parkinson's disease. This finding suggests a common pathological pathways among these seemingly distant neurodegenerative conditions. Building on our findings, we then compared these pairwise transcriptomic similarities with pairwise shared heritabilities, which was estimated from genome-wide association studies. This allowed us to explore the genetic underpinning of our observed similarities. We discover high concordance between co-heritability and transcriptomic similarity. This finding suggests that genetic risk factor might be shared among traits exhibiting similar transcriptomic profiles. When we performed the same analysis using cell type specific manner, we found that chandelier cells exhibited highest concordance between co-heritability and transcriptomic similarities. This highlights a critical role for this neuronal subtype in a shared genetic architecture of these complex brain disorders. Next, we directed our focus to transcriptomic variation associated with Alzheimer's disease pathology. Through composition analysis, clinical variable measuring AD severity, we were able to nominate a specific cell type that is a crucial player in AD pathology. To tease apart complex interplay, we employed a series of mediation analysis. This approach allowed us to decipher a causal relationship between changes in a cell type composition and a progression of AD pathologies. Our finding revealed a distinct role for 2 interneuronal subtypes. We observed a mitigating effect of somatostatin interneurons on amyloid pathology, suggesting may protect against amyloid accumulations. Conversely, we found an exacerbating effect on LAMP5 interneurons on dementia severity, indicating that LAMP5 interneuron might contribute to cognitive decline in AD. These highlights complex cellular mechanisms influencing AD progression. While traditional linear model can tell us the size and direction of the disease effect, they often fall short of revealing nonlinear dynamics that unfolds over the course of the disease. To truly understand this complex pathological dynamics, we employed a neural network approach. This allowed us to infer intricate disease trajectories using a semi-quantitative measure of AD severity. Specifically, we inferred 2 distinct trajectories, one based on tau proteinopathy and the other using dementia severity. By integrating these 2 perspectives, we gain a much more nuanced and characterization of the disease progression. Then organize this disease course into 4 distinct modules, each representing a unique pattern of change. Interestingly enough, our analysis revealed the late increasing trajectories showed a stronger association with the AD genetics, and further stratification allowed us to differentiate between early and late changes. And we're finding evidence of early activation of immune function and damaging changes in the vascular functions, which implicated the cognitive decline and disruption of blood-brain barriers. To summarize, we generated one of the largest single-cell cross-disorder atlas on a human prefrontal cortex. Our robust taxonomy allowed us to tease apart shared and distinct component of the multiple human brain disorders, further characterizing AD pathology using these disease pseudotime revealed early immune activation and damaging changes in the neurovascular functions. This study offers foundational framework for targeted cross-disorder treatment that address molecular complexity of brain dysfunction. With that, I'd like to extend my sincere gratitude to all the collaborators who contributed to this extensive work over many years. I also want to acknowledge generous funding and vital support from funding agencies, including NIA and brain banks. Thank you.
All right, thank you so much, Donghoon. We're going to keep the questions for the end. We're going to have a Q&A session. So we're going to move to our next speaker.